Okay. Good morning, everyone. It's right at 11, so I'll begin, but continue to let people join for the next minute or so. So I'm the host today for our Science Communications webinar, and I want to welcome y'all to our December edition. As usual, we're going to ask that you keep your cameras off and your microphones muted as we go through the webinar. And as you have questions, go ahead and put them in the chat. And at the end, we'll have the opportunity to go over the questions together after the presentation. But today we have Dr. Fred Scharf. He's a professor in the Department of Biology and Marine Biology at University of North Carolina at Wilmington. He's been in that position at UNCW since 2002, primarily working with marine fishes, but most recently with anadromous fishes. And he's going to present information on his research that focuses on the Atlantic sturgeon in the Cape Fear River. And today, the title of his presentation is Atlantic Sturgeon in the Cape Fear River, Evidence for Spawning Populations and Migratory Dynamics Across Stages. And so with that, I will turn it over to Dr. Scharf, and I'll close my video out and let you have the screen. Thank you.
Okay, thanks, Sarah. Appreciate it. So it's really nice to be here and share some of our recent findings with folks. As Sarah indicated, I plan to give you guys an update on the work that we're doing on Atlantic sturgeon, primarily in the Cape Fear River, that we started in 2021. And so most of you guys are familiar with Atlantic sturgeon, that we have had historic populations of Atlantic sturgeon along the east coast of the United States, and that most of those populations have been impacted by overfishing and habitat loss that have caused tremendous population declines, most notably in the late 1800s and early 1900s. Then they underwent sort of a long period of sort of stasis or very low landings throughout most of the 20th century. And it wasn't until 1998 that the Atlantic States Marine Fisheries Commission issued a moratorium on harvest along the Atlantic coast. And since then, they've been monitored and studied to try to understand recovery patterns. They were listed federally by NOAA as endangered in 2012, and the management structure divides sturgeon up into 5 what are called distinct population segments, which you can see on the map of the Atlantic coast. And where we are in the Cape Fear River is part of what's considered the Carolina DPS, which includes the Roanoke River south to the Cooper River in South Carolina. And the Cape Fear River sits sort of in the middle of the Carolina DPS. The 4 DPSs from the South Atlantic, Carolina, Chesapeake, and the New York Bight are all listed as endangered, and the Gulf of Maine DPS is listed as threatened. And so there's been some work done in several of the North Carolina rivers, most notably the Roanoke and the Cape Fear historically, and indicating, particularly some of the most recent work in the mid part of the last decade, between 2012 and 15 or so, indicating that there was a good possibility that we had a viable extant population occupying the Cape Fear River. In addition, there's been some work that has confirmed that we have spawning population in the Roanoke River at the north end of the northern edge of the DPS. And so beginning in 2019, 2020, we started to have some conversations with the Sturgeon Recovery Coordinator for the Southeast, and they asked us if we would be interested in doing some work in the Cape Fear River. And so with the help of Jeremy McCargo, who's the anadromous fish coordinator for the Wildlife Resources Commission, the work we are on the Cape Fear is really focused on a couple of areas. The first is to try to confirm that we actually have a spawning population in the Cape Fear River, and I'll go through on the next slide some of the evidence to support the existence of a spawning population. The second was to try to identify where the spawning was likely occurring. And then the third objective related to spawning was to look at some of the behavior in terms of immigration, emigration of adults, the timing of those events, pathways that they used, and any particular habitats that might support the adult spawning aggregation. So in terms of the evidence to determine an extant spawning population, on this chart, if you go from the bottom to the top, the evidence increases in the strength of that assertion. And so the capture of adults in a river system, but not in the spawning habitat, would be still an indication of uncertain spawning population, the presence of a spawning population in the river, because the adults have been shown to stray into other rivers occasionally outside of the spawning season. The capture of males in spawning condition, actively releasing milt in what is suspected at least of being a spawning habitat, would be another criteria which would indicate a strong possibility that they're spawning in the system. The next two categories, the presence of gravid females in the spawning habitat or the presence of river resident juveniles within the system, would be near certain indicators of a spawning population. And those river resident juveniles are fish that are generally less than 2 years old, that are thought to remain in their natal system up until that age. So fish, if you capture fish in that age range, the assumption is they were spawned in that river. And then the strongest evidence is the presence of eggs that can be confirmed as Atlantic sturgeon, or actually observing actively spawning females with milting males in the spawning habitat, which can be done in some systems when they spawn in clear, shallow waters. And so I'll hit on these a little bit later in terms of what we've been able to obtain so far in terms of trying to meet some of these criteria for a spawning population. In addition to adults, we wanted to monitor the movements and habitat use of subadult fish. So these are fish that are not yet mature, but are beyond the juvenile stage and tend to show lots of mixing within DPSs and even across DPSs. So we have fish within these life stages that can move into rivers across different states. And then lastly, the juveniles to monitor, those river resident juveniles to monitor seasonal movements. And if we could estimate abundance using mark recapture, which requires capturing lots of those individuals, we would try to see if we could develop a recruitment index for the Cape Fear River. These two life stages have some particular importance because they tend to spend a lot of time in the river. The river resident juveniles spend almost all year round in the river, and then the subadults typically, depending on the latitude, anywhere from 4 or 5 up to 7 or 8 months a year in the river systems before they move back into the ocean. So impacts of activities in the river are most acute on these life stages. The adults tend to be here for much shorter periods of time. So in the Cape Fear River, the lower Cape Fear River, which extends from Bald Head Island at the interface with the ocean up to Wilmington, which is located about 40 or 50 kilometers upriver. And then it branches into the Northeast Cape Fear branches off to the northeast, which is a large tributary. And then further up, we see the Black River branch off to the north. And then the main stem Cape Fear River, which runs up towards beyond Fayetteville and eventually the confluence of the Deep and the Haw Rivers, where the Cape Fear is formed. We've been focused on the capture of fish using short soak gill nets. The adults, we use large mesh gill nets. The subadults and the juveniles, we're using smaller mesh gill nets. The soak times are all dictated by water temperature and oxygen saturation. And then the standard processing includes taking secondary fin rays for aging, taking pelvic tissue samples, pelvic fin tissue samples for genetics, pit tagging fish, and then if fish meet certain criteria, we put acoustic tags in. We have an array of acoustic receivers that has been established. We were doing some work on flathead catfish movements in the river. We've had arrays in the river for that work off and on since 2018, but established fully since 2020. We've had a network of acoustic receivers up the northeast Cape Fear, which you can see going up towards Kenansville, up the Black River towards Ivanhoe, and then up the main stem Cape Fear River, up beyond Lock and Dam 3. We've since brought some of the receivers in the Black and the northeast down a little bit. We don't go up quite as far anymore because our flathead work is finished, and we haven't seen any evidence of sturgeon going up that far. Or any other anadromous fish in most cases. But we've expanded our coverage in the main stem Cape Fear. We have a lot more coverage around the locks and dams. The first lock and dam is located at about river kilometer 97, so almost about 100 river kilometers from the ocean is where we have the first impediment to migration. And we've targeted our adult collections there as the first barrier to a migrating adult. And so this is just a figure of what we've captured so far in terms of just length frequencies. So this is just length this is fork length on the x-axis and numbers captured on the y. The adults are out here on the right. And so the NOAA biological opinion designates an adult at 1,300 millimeters fork length, so 1.3 meters and above are adults. The subadults technically, according to the NOAA biological opinion, range from 1,000 to 1,300, and from about 500 to 1,000, this is considered what's called the marine migratory juvenile phase. So they're migrating into the ocean periodically throughout this phase, and then they move into a subadult phase here and then become adults after 1,300. 500 millimeters is an approximate cutoff for the river resident juveniles. We use that. It's probably a bit conservative. We think some of these fish that are 520 to 530 are probably still river resident juveniles, depending on what time of year they're caught. And we've captured quite a few of those fish as an indicator of the presence of a spawning population. And as you can see here in the blue, we've captured quite a few of those river resident juveniles. The majority of the fish that we capture are these marine migratory juveniles, and we've captured fish from multiple states so far. So we've detected fish that had acoustic tags that were tagged in multiple rivers in Georgia and South Carolina, from the PD, the Edisto, the Savannah, the Altamaha River. And we've also captured fish that had pit tags that were originally tagged in those systems. So lots of evidence for mixing across the rivers in the South between both the Carolina DPS and the South Atlantic DPS. And I'll touch on that at the end in terms of some of our current work and future directions. In terms of the length at age pattern, so this is just fork length as a function of age. The maximum age so far that we've seen is 15. So you can see our adults that are over 1,300, and they ranged anywhere from 9 to 15. So we haven't seen anything in our system yet beyond age 15. In the southern rivers, I think there's been fish aged up to 25 or so. There's a latitudinal gradient in longevity. So the populations up in Maine, fish may live to be 50 or 60, maybe even more. There's some thoughts they might even live older than that. And then, but the fish in the southern part of the range grow faster, have shorter lifespans. We think more in the order of 25 to 30. But we haven't seen anything yet older than 15 in the Cape Fear. So far we're seeing linear growth. We haven't really seen asymptotic growth yet because we haven't gotten to some of those oldest fish yet. In the James River, they also saw linear growth up through these size ranges. Once you start to see some older fish, you'll start to see some asymptotic growth at the high end, but we haven't caught those fish yet. So I'm going to show you some examples of some of the residence and emigration patterns of some adults that we captured. So recall our first season of sampling adults was in the spring of 2021. Now, I'll preface it by saying in the north, in the Chesapeake and above that, primarily we see spring spawning, tends to be late in the spring, gets warmer much later up there. So a lot of spawning in May, June. But in the James River, they've identified distinct spawning cohorts that spawn both fall and spring, and those have been distinguished genetically. Similar to that, we've seen that below the Cape Fear in the Edisto River, where we have fall and spring cohorts, and that's been noted in the PD as well. But as you get further down into Georgia, most of those rivers only show evidence of fall spawning and no spring spawning. So we started in the spring, and we focused on looking for adults in the spring. And that first spring in 2021, we captured 5 adults. They were all males. They were all actively releasing milt, and they were all captured just below Lock and Dam 1, within 2 or 3 kilometers of that dam. And they were tagged up here. Some of those individuals, depending, the first fish we tagged was April the 8th. The last fish we tagged was around April 22nd or 23rd. I think we might have tagged one. I think we did tag one April 30th maybe, and it immediately left. Some of the fish we tagged late April left pretty quickly after we tagged them. Everyone was gone, had emigrated out of the system by early May. And so the last fish that we saw pass below Sutton Lake, which is just above downtown Wilmington. We never saw any fish that went below that ever came back above that. So when they passed that point, they were emigrating, and we didn't at that point in the spring of 21 have as much receiver infrastructure in the lower river down towards Bald Head to detect their actual emigration timing. But we detected them moving past Wilmington, as by early May they were all gone from the system. Now in the following year, in the spring of 2022, we saw 3 of those fish. These 3 colors here, blue, red, and green, are 3 different individuals. 3 of those fish that we tagged in 21 returned in spring of 22. And so you can see their emigration timing, and we see some of this exploratory behavior where you'll see a fish come into the system in February, and this fish came all the way up to Wilmington and then moved back down toward the inlet, stayed down there for roughly a couple of weeks, and then moved all the way up to Lock and Dam 1 very rapidly, stayed there for a fairly long period of time, about five weeks or so, and then emigrated from the system. We also see some variation in entrance timing. Again, this fish showed up as early as sort of mid to late February, and these other fish came in much later in mid-March or so, was with their first detections in the system, and differences in sort of duration and timing within the river. And then and then eventual emigration from the system. I'm hoping that I just got a message from, oh, that's an old message. Okay. So I'm good. I should be good. Let's see. So hopefully this animation works. So there's an animation, you can see the time up above. This is late February. That first fish that came in, moved up, came back to the inlet, and then stayed there for a couple of weeks. And so now we're into the first week of March. And then you'll eventually see this fish start to move up the river, and it moves up. And this area up here in the upper left of that plot is Lock and Dam number 1. So he's up there, it's mid-March or so. Eventually you see another fish come in. Fish spent a long time staging in the lower river below Wilmington, was passed by another fish, and then eventually moved up. In both cases, the fish in blue and this fish in red that'll eventually move up here nosed into the Black River for very short periods of time, usually just a day or so, figured out that it wasn't where they wanted to be, and then eventually worked their way up to Lock and Dam 1. And you can see them milling around Lock and Dam 1, and then they decide to emigrate. The red fish is leaving, gets passed by this green fish. They both were gone by April 21st or so. This individual stayed up there for another 3 or 4 days and then left. And then the video starts over again. And so you see, entry as early as February, but you can see some entry, departure, and re-entry. So some uncertainty about whether they're ready to go to Lock and Dam 1. You see some brief entry into the Black River by a couple of fish, but it's very short. Some staging, and then some variability in those residence durations and the timing of their exit. So those same 3 fish, those same 3 males came back again in the spring of 2023. And so one of the fish we only heard very, very briefly, and then we didn't hear it again. And I'm not 100% sure right now of the fate of that fish, whether that fish has been detected in the ocean since then, or whether it may have suffered a mortality event in the river in late March of 2023. And so there's always a lag in terms of the ocean detections that we get from the Matzos and the FACT network along the coast, because it depends on other researchers uploading their detection files in a timely way to the system, and then they get downloaded, usually quarterly, every 3 to 4 months. And so I'm not 100% sure right now what the fate of that fish is, but it only showed up for a short period of time. One of the other fish, though, which is the same fish that did this the previous year, sort of nosed in, a lot of indecision in late February, came all the way up to Wilmington, back to the inlet, back to Wilmington, back near the inlet. Finally, around the middle of March, around March 20th, it decided to go all the way up to Lock and Dam 1, stayed up there for 10 days or so, went all the way back to the inlet again, and turned around and went all the way back to Lock and Dam 1 again, stayed there for another 2 to 3 weeks, and then emigrated the system. Whereas the individual in red showed up in early April, went directly to Lock and Dam 1, stayed up there for 3 or 4 weeks, and then emigrated from the system. Now, this behavior of being at Lock and Dam 1 for a couple of weeks, emigrating, and then returning very quickly wasn't an isolated incident. We saw that again the following year. And so this is the data from the spring of 23, just this past spring, for fish that we tagged in 2022. And this includes a female. I'll get to her in a second. But the orange and the red and the deeper brown here are 3 different males. And you can see this male that we tagged in 22 that came in very early February, went all the way up to Lock and Dam 1, was up there by, the middle of February, stayed up there for a very long period of time, and then went all the way back to the inlet around the end of the first week of April, and then quickly returned and came back to Lock and Dam 1 again for a period of time before eventually immigrating. And so we don't really know what triggers that kind of behavior, where they've been at Lock and Dam 1 for a fairly substantial period of time, emigrate to the ocean, or at least back to the inlet, and immediately turn around and go back to Lock and Dam 1. We suspect that they may have encountered a female, and the female was on her way to Lock and Dam 1, and so they turned around and followed her back to Lock and Dam 1. But that's just speculation. This is these other two males came in later in March after a small amount of sort of exploratory behavior, moved up fairly quickly to Lock and Dam 1, stayed there, and emigrated kind of close together within a few days of each other in early May. This is the female fish in brown in the middle that we tagged in 2022. Now it's notable that she returned in 2023, because most of the observations that have been made in the Mid-Atlantic New York Bight indicate that the females don't spawn every year. And most of that is based on the return patterns of acoustically tagged females, and they see them come back on 2 and 3 year cycles. They don't tend to come back each year. So she did come back in 2023. She was captured in 2022. While we were tagging her, we confirmed that she was gravid with eggs. And then she emigrated the system, about, a week or so after we tagged her in 2022, along with most of the other males. And in 23, she returned. And so there may be, again, a latitudinal gradient to the spawning frequency, where in the southern regions, we may see more consistent annual spawning by females. So we're anxious to look this spring in 2024 to see if she comes back again. But what you can see in 23 is that she also went up to Wilmington, came back down near the inlet, went back up to Wilmington, came back down near the inlet, went back up to Wilmington, stayed there for a week or so, finally went up to Lock and Dam 1, and only remained resident at Lock and Dam 1 for about 10 days before she left. So she spent a lot more time in the lower river than she did up at the spawning grounds, which puts her at much greater risk for human interaction, primarily due to shipping. All the shipping traffic is in this part of the river here, and dredging activities and commercial fishing operations in the lower river. All of that stuff occurs from Wilmington south, not nearly as much of it above Wilmington. So she spent a lot of time in the risky part of the river before she made it up to the spawning habitat. This is just her by herself, so it's easier to see what she did in terms of this movement in the lower river. And this extends from about March 18th or 19th all the way until, about April 4th or so. So she spent, the better part of, 2.5 weeks in the lower river, and only spent about 1.5 weeks in the upper river before she emigrated. We have a really great collaboration that we're doing in the river with Troy Farmer and Aaron Bunch at Clemson. So Troy is a professor of fisheries at Clemson, and Aaron is working on a PhD under Troy's supervision. And they had some funding from the Army Corps to look at fine-scale behavior of anadromous fish below the lock and dams in the Cape Fear River. And so this is data from the Lock and Dam 1. that just shows the density of detection. So they had in the spring of 21 and 22, they had an array of fine scale receivers, both above, below and above Lock and Dam 1. And these were high resolution receivers that could overcome the turbulence below the dam and hear these fish tags. And so they showed that the area and so they put some tags, some of these high resolution tags in Atlantic sturgeon in the spring of 21 and 22. This is data for the nine adults that were up there in the spring of 22 that had these high frequency tags. And you can see the density of detections. This is the fishing pier at Lock and Dam number 1. And so there's a deep area right off the fishing pier where it's about 30 feet deep or so. And this is where the most of the detections were. And we're working with Aaron right now. Jake, my student, is working with Aaron now to quantify some of these behaviors in terms of the frequency of adult visits to this area here, the duration of time that they spend in here, how many visits they make in here. So we, from our broad scale acoustic arrays, we have, the residence time patterns. So we know when they're up in that area, but then the fine scale data can tell us when they're actually in this area here where we suspect the spawning is actually occurring, and how many entrances and exits that they make from that area, and whether there's any consistency in that behavior, both within individuals and across fish. So I mentioned in the beginning that there's evidence for fall and spring dual spawning in rivers both north of us and south of us. So we assumed coming in that there would be a good likelihood that there was both a spring and a fall spawning cohort in the Cape Fear River. So far, we've only found very limited evidence of the presence of adults in the fall. In fact, we've only caught one. We haven't put as much sampling effort in the fall as we have in the spring, so we can't say that sort of unequivocally. But in the effort we have put in, which we made some efforts in the fall of 21, and then Jake made a lot of sampling effort at Lock and Dam 1 in the fall of 22, and he only caught one male that he tagged below Lock and Dam 1 in late September of 2022, and this is that fish. And one of the things he noticed is that the ventral scutes were really beat up. You can see they're really red. There's a lot of scrapes and cuts along the ventral scutes. And so he wondered if this fish had actually migrated above the rock arch at Lock and Dam 1, the fish arch, the swimway, and then he caught it after it had come back down. But he had no way to know because it was captured below. He tagged it, and it milled around in the Cape Fear for a while and eventually emigrated, and it never went back above the rock arch. That fish returned this fall. He detected it, and it did pass Lock and Dam 1. It did swim over the rock arch in late September this fall. It only remained above there for a few days, and then it came back down below Lock and Dam 1 and then stayed in the system into October and eventually emigrated. So we've never seen, I should preface that by saying we've never seen a spring fish. None of the fish we've tagged in the spring, adults that we've tagged in the spring, which is now over 20, we've never seen any of the spring fish move over the rock arch at Lock and Dam 1. But this fall fish and moved above the rock arch in the fall of this fall of 2023. And it's interesting because the folks in South Carolina just published a paper recently on the fall and spring spawning cohorts in the PD River. And one of the things that they noted in that publication was that the fall fish migrated much further up the river, 40 or 50 kilometers further up the river than the spring fish did. And it was fairly consistent. And it's much warmer in the fall. And so they hypothesized that those fall fish may need to get further up the river to where some of the flow and oxygen conditions are better because the temperatures are higher. When they're in there in September, you know, in the up here at Lock and Dam 1, the temperatures up here are still in the high 20s. Whereas when we're catching fish in the spring, they're in the low 20s. So it's 5 to 7 degrees warmer up there during the what we suspect is the fall spawning season. So we're continuing to look. One of the other criteria for evidence of a spawning population is the presence of eggs. And so we started to do some egg mat sampling in the spring of 22 and 23. And we were really lucky in the spring. In April of 23, we collected 3 eggs that we suspected were sturgeon. We sent those to the USGS genetics lab up in West Virginia, and they applied a very specific primer that was developed to identify those eggs, and sure enough, they've been identified as Atlantic sturgeon eggs. And so it represents the most definitive evidence that we have active spawning in the Cape Fear River is the collection of these eggs. And if you look close enough, you can almost see the dorsal scutes on this individual inside the egg case. I mentioned beyond the confirmation of adult spawning population presence in their system that we are interested in looking at movements of the younger life stages, since they spend a lot more time in the river and potentially a lot more time in the parts of the river where they'd be at risk for sources of mortality from ship strikes, dredging operations, fishing operations, etc. So this is just a summary of some river resident juveniles. It's about 8 individuals that we put acoustic tags in. 4 of them were tagged in December of 21, and then the others were tagged in the late spring and fall of 22. And one of the things that you notice is that some of them emigrated or showed some evidence of emigration to the ocean, but many of them did not. And the evidence for that emigration is interesting and size dependent. And so I'll show you a few examples. So this is the area in the river that's highlighted here. Wilmington's right about in the middle of this circle. This area in the middle of this highlighted circle is about where the salt fresh interface typically is. It changes daily with the tide, but it also changes with precipitation. Sometimes it can shift upriver 8 or 10 kilometers and sometimes downriver, depending on how much rain we get. But the movements of these river resident juveniles, this is a fish that was tagged in December, they tend to spend a lot of their time in this circle. And we see a little bit of evidence of movement. This is river kilometer on the y-axis, downriver during the coldest months. So that first winter in late February, we saw this fish move down to river kilometer 20, but it never left the river. And then the red circles are when it was up in the northeast Cape Fear, but it's still in the top of this circle up here. In the next winter, that fish moved down in late December, in December 2022. Now, if you guys remember last winter, that week of Christmas was the week we got the coldest weather of the year. And it aligns with when this fish moved down river toward the ocean. It got down as low as river kilometer 10. So it was within 10 kilometers of the ocean inlet. It may have gotten closer. Our detection probabilities down here are less. It's just a more dynamic environment to hear fish. And we lose receivers a lot down there. So it may have gotten closer to the inlet. But this aligns with some evidence that was observed in the Savannah River, that these river resident juveniles, while they tend not to leave the river, they move downriver toward the ocean when it's cold. Here's another one that was tagged in the spring, spent a lot of its time in the northeast Cape Fear River, around river kilometer 50, but it also moved down all the way to the ocean inlet in December of 2022, around that same time period when it got really cold around Christmas time. So in terms of the river resident juveniles, in an overview, again, they tend to aggregate near the salt fresh interface, generally between river kilometers 30 and 60, but a lot between 45 and 50, right around Wilmington, right around downtown Wilmington. The emigration to the ocean appears to be size dependent. The 3 individuals that were bigger than 500 millimeters emigrated. The fish that were smaller did not. They did show downriver movements in the winter, and the timing of emigration was really variable. And so far, those fish that emigrated, we haven't seen return from those fish. So they've taken up spots in other rivers and haven't come back yet. In terms of those marine migratory juveniles that are a little bit older, right? This is a bunch of fish that were tagged in either 21, most of them in 2021, either in the spring or in the late fall, early winter. And the main thing to notice is the emigration date. Almost all of them emigrate, right? And most of them in November. So all the ones we tagged in May and October, they all emigrated in November. We tagged some fish later, and those fish emigrated later. They were still here in December, these 4 fish at the bottom. I think that, yeah. These 4 fish at the bottom all emigrated later, where the fish highlighted in green all emigrated much more similarly. And many of those fish returned the following spring, most of them in late March, some of them in mid-April, but in between late March and mid-April, they all came back. And many of them showed those same emigration patterns the following year. In fact, all the ones that emigrated late, these 5 fish at the bottom, they all sort of got on that emigration train much earlier the next year. And so you see these sorts of patterns where, again, they hang out a lot near the salt fresh interface. These red dots are the northeast Cape Fear, just above Wilmington, that first branch. And then you'll see this emigration period at the bottom, where in the late part of the winter they disappear and then they reemerge. And then they go right back to that salt fresh interface, spend most of their time there, and then emigrate again. This is another fish that showed that same pattern. Emigrates, comes back in, spends most of its time in that salt fresh interface around river kilometer 50 to 60, and then a fairly abrupt emigration. And one of the things we saw with this life stage is the emigration seemed to be triggered by rapid and fairly considerable drops in temperature. So in 2021, the primary window of emigration occurred immediately following a 5-degree temperature drop that occurred from November. You know, the emigration was between November 6th and the 22nd. During that 2-week window, roughly, was when most fish emigrated. And the week or so before, we saw a 5 degrees Celsius drop in temperature. The same thing we saw in 2022, where most of the fish emigrated between the 16th and 29th of November. And right in that window, we saw another 5-degree drop in temperature. So when we go from 20 down to 15 or so, when that first hint that winter is coming, most of those fish leave. And we've heard a lot of those fish off of Charleston. So the South Carolina DNR folks have lots of receivers outside of their rivers in the coastal ocean. And we've heard a lot of our fish have been picked up on their receivers. So it seems like the marine migratory fish that are in the Cape Fear during the warmer time of the year, they go south in the winter. We haven't seen them show up on receivers up in the Roanoke or rivers to the north. They all seem to go south. So they tend to have a little bit more use of upriver habitats. They'll stray a little further up, maybe up toward river kilometer 70. Some of them even have nosed into the Black River for short periods of time, the lower Black. They don't go up the Black very far. Again, most of them, nearly all of them emigrate. They don't overwinter in the Cape Fear, and they're usually gone for 3 or 4 months. And then when they return, they tend to return mid-March to late April, and that return timing tends to be very consistent for these life stages. So this is just a summary of what we've done so far. There's a lot of text up here, but you know, we've got 20 adults that we've tagged spring 21, 22, 23. It includes a gravid female. We've had males that have come back 3 years in a row. In fact, one of the males we recaptured one this spring that we tagged initially in 21, and it was good that we did because we needed to put a new tag in him because the original tag wasn't working anymore. We're working with the Clemson folks on some of the high resolution data, and Jake is working on quantifying more information on timing and pathways during immigration and immigration. Lots of subadults captured, many from other places. Again, lots of river resident juveniles have been captured, which is strong evidence of spawning in the system. And we're trying to use the seasonal movement data of those river resident juveniles to try to identify mortality risks throughout the year. And again, we have eggs collected, and the application of those primers have confirmed that they were Atlantics. So if we look at the evidence to determine spawning, you know, we've certainly captured adults. We've captured milting males in what we think is the spawning habitat below Lock and Dam 1. We've caught a gravid female. We've caught river resident juveniles in all 3 years, and we've got the presence of eggs, of Atlantic sturgeon eggs. We can't see active spawning females because they're spawning in deep water and the Cape Fear is too turbid. But we have all the evidence we could possibly have that indicates we do have an extant spawning population in the Cape Fear River, at least in the spring. And the future work, we want to see if we can capture more river resident juveniles to see if we can do mark recapture. We captured a lot in the spring and early summer of 21, and so we anticipated moving forward that we would be able to catch those same numbers. And the environmental conditions just have made it challenging. It's caused the salt fresh interface to move into places where it's harder for us to catch fish. So we haven't caught enough numbers in the spring and early summer to conduct mark recapture to get an estimate of abundance yet of the juveniles. Again, more acoustic. I think our adult work will continue every spring just to continue to monitor the fish we have tagged and continue to get more tagging and genetic information from adults. We're going to continue to look for evidence of fall spawning. And then we have a new NOAA-funded project in collaboration with the folks in Georgia and South Carolina to look at mixing rates of these marine migratory juveniles, subadult stages, across the rivers in the South Atlantic. And so that was based on a lot of the patterns we were seeing in 21 and 22, where all of the fish that we captured that weren't from the Cape Fear, they were all from Georgia and South Carolina. And so we've started a combined acoustic tagging and genetic assignment project where we're going to look at the extent of mixing across multiple rivers in these 2 DPSs to see if we can get a better handle on whether certain rivers serve as important aggregation sites. What's the extent of mixing? Is there more straying within rivers that are close by, or are fish moving large distances across different rivers? And especially whether many of the subadults that we catch over and over again in the Cape Fear are those natal fish to the Cape Fear. The adults and river resident juveniles we have are hopefully going to inform a unique genetic signature for the Cape Fear. So then when we catch the migratory juveniles, we should be able to see if they're assigned to the Cape Fear or if they're coming from another system. So that's the work we've just started with the folks in Georgia and South Carolina. And so with that, I'm happy to answer questions for folks on what we've been seeing so far and what our plans are moving forward.
Mm-hmm. Yeah, thank you, Dr. Scharf. We're open for questions. I'll look for questions in the chat. In the meantime, I have a quick question. I'm curious about the mortality rate of these sturgeons at different size classes. I assume once they get to a certain size class, the mortality rate is much lower.
Well, I think certainly from a, from natural sources, yes. But I think most of the threats are not natural. In many of the systems in the Chesapeake Bay and Delaware Bay, it's ship strikes, and so a lot of it just depends on the extent of time that individuals spend in those shipping lanes. The problem is they use the deep channels, which are the channels that the ships are using, and in many cases the ships, there's very little room under the ships for sturgeon. So in Wilmington, for instance, the main channel is maintained at about 42 feet in depth, and many of the ships that use it are drawing 36 to 37 feet of water. So there's only 5 feet under the ships. The other is dredging activities that occur in the lower river for both navigational channel maintenance, but also some of the coastal ocean dredging that occurs for beach renourishment projects. And those tend to occur in the winter, which is when the sturgeon tend to be in those habitats.
Yeah, I was wondering about that too. So we have a couple other questions. One from Heather is asking, by the fish that were tagged, then they immediately immigrated. Do you think that the tagging procedure contributed to their behavior at all?
It's possible, although we haven't, we've seen that mostly with fish that we tagged late. And so for we've tagged some adults that we tagged them, captured them later in the season, and they tend to leave fairly quickly, but it's also when many of the other fish are leaving. So it's possible. It's hard to separate sometimes. We haven't really seen that behavior for fish that we've tagged early in the spawning season. They tend to stay.
Okay. Yeah, and then Jeremy McCarter has a great question. What impact do you think Lock and Dam 1 is having on spawning success of ATS during spring and fall? Yeah, I was kind of curious about that too, if that was having an impact on the fish behavior and spawning.
Yeah. I mean, as I said, I think we have all the evidence that we could have, that we have a spawning population that's active in the Cape Fear River. We don't know yet. I think one of the next steps for us too is to try to combine some side scan sonar abundance estimations, right, that have been used successfully in some of the other rivers. There's a couple of recent papers published in the Savannah River where they've estimated the size of the adult spawning population using side scan. So we may try to, the folks that we have a good side scan system, but the folks at the community college here in Wilmington that run the marine technology program, they have a much more sophisticated side scan system. So we may try to work with them to see if we can do some scans during the month of April in combination with our acoustic tagging to estimate the size of the spawning population in the river. It seems like they're successfully spawning below Lock and Dam 1 in the spring. We haven't seen, any of the 20 plus tagged adults that we've tagged in the last 3 springs. None of them have gone above that dam. And, the fact that the dams themselves have been there for roughly 100 years, I start to think about how quickly we should expect the fish to adapt to the rock arch. Certainly this one fall fish managed to get over that rock arch. So it can be done because it went across in 23 and the modification was completed in the fall of 21. So since spring of 22, there's been some pathways that should promote more movement of fish across it. And so they can get across it. I just wonder how motivated they are to get across it if they're successfully spawning below it. I don't know. Same with some of the other species. I'm not sure. It may be more important for some than others. It may be that in the spring, the sturgeon are have been able to be very successful below Lock and Dam 1, and they may continue to be.
Yeah, interesting. Well, we have a few minutes left, and I didn't see any more questions. I appreciate you coming and giving this presentation. This is really interesting work.
Yeah, and I want to, I really want to acknowledge the help of Jeremy and the staff of the Wildlife Resources Commission, because the NOAA funding that we have is it has to pass through a state agency, because the grants are species recovery grants that are made to the states, and the leadership at WRC has been really helpful and supportive, and basically just passes those budgets straight through to us, and is really helpful in administering those for us, and helping make sure that we meet all the reporting requirements and other things. And so we couldn't do it without them. And I know Jeremy's going to come out with us this spring, so that he can wrestle one of the big ones for us.
Yeah, that would be pretty cool.
We need some additional muscle on the boats, because those fish are big, not quite as big as the one in this picture, but
Yeah, Jeremy asked, should we be looking for spring spawning fish lower in the Roanoke?
Yeah, that's a good question. I know that that Matt's been trying to tag spring spawning fish, and he's mostly been trying to catch them on their way out, which is the one nice thing about Lock and Dam 1 in the Cape Fear, right, is it creates a barrier. So it creates this bottleneck where we can sample and have high efficiency in catching adults. In the Roanoke, our colleague from Virginia, Matt Belazic's been sampling, but he can't sample that much up near the spawning grounds, because he doesn't want to impact the striped bass fishery that's happening, because you have migratory striped bass that are using those same habitats. So he's mostly been fishing much lower in the Roanoke, just trying to intercept fish on their way out. And I think that he's caught a few, but I wonder if they may not be going up as far, and they don't, they could spawn in locations further south, and they don't need to go up to Roanoke Rapids like they do in the fall, right? Because that's where the fall spawning occurs. He's definitely caught adults up there in the fall, and egg sampling has been done up there in the fall in that area near Weldon and but in the spring they could be spawning lower. It's possible for sure.
Yeah. Well, we're about at time here. I do want to note Director Ingram says he can't provide the mussels, but he wishes to be part of the tagging in the spring.
Okay.
So we'll keep him in mind for that. Thanks very much. And thanks everybody for your participation and questions. Our January presentation is going to be given by John Levitt and Anna Stefanovic, and they're going to talk about lots of our GIS projects across the agency. So we'll have them in January. So thank you very much. I appreciate it.
Good to see you guys. See you, Sarah.
Take care. Bye-bye.